Hi, Chemistry 3111. This is chapter 25, wherein we discuss amino acids, peptides, and proteins. And of course, peptides and proteins are made from amino acids. So just by way of introduction, some things you might want to think about reviewing before we get into this chapter are designating RNS configurations at a chiral center. So those are covering the Kahn-Ingold Prelog rules, and that's way back in chapter 5, which is the stereochemistry chapter. Structure and reactivity of carboxylic acids. Obviously, we're not going to look at every reaction that we covered in that chapter, but a couple of those reactions might come up in this chapter. Nucleophilic acyl substitution, and then finally, properties of amines. Knowing that an amine can behave as a base and as a nucleophile will be handy in this chapter. Let's take a look at amino acids. It says that proteins are nothing more than polymers of amino acids. Now, even though we haven't covered the chapter on polymers, I'd wager that you know enough about polymers to know that they have repeating units, right? If you take something like saran wrap, which is a polymer, or polyethylene terephthalate, which is a polymer, it just has a repeating unit that just repeats and repeats and repeats. Well, you've heard a lot about proteins, I'm sure, in your biology classes, and proteins are nothing more than polymers, but they're naturally occurring polymers, and the monomers that make up proteins, again, which are polymers, are amino acids. So an amino acid, just like the name sounds, it contains an amino group, which is an NH2 group, okay? And it contains a carboxyl group, which is where we get a carboxylic acid. So we have a base and an acid in the same molecule, which is pretty interesting. And the amino acids of primary concern to us in organic chemistry too are what are called the alpha amino acids. It says that the amino and carboxyl groups are only separated by one carbon. If you think about carbonyl chemistry, and we looked at plenty of carbonyl chemistry, let's say you had some kind of aldehyde like this, for example. You have the carbonyl carbon, which is this one here. Well, you said that the carbon next to it is the alpha carbon. Following that is the beta carbon. After that, you have the gamma and the delta, so on and so forth. But the idea is this: in an amino acid, we do have a carbonyl in our carboxyl group, and the carbon that is next to the carbonyl carbon, that is the alpha carbon. So this one highlighted in green, this is the alpha carbon of an alpha amino acid. Now, what does the alpha mean? Look, if I have my carbonyl, and I'm going to draw it over here to try to clear things up. Here's my carboxyl group. This is my alpha carbon, and I have an amine attached directly to it. Again, this is my alpha carbon, so alpha amino acid. And then you can see that there's an R group also coming off of the alpha carbon as well. So that's an alpha amino acid. And you can see that that alpha carbon, if the other group on here is a hydrogen, if the R group is anything but a hydrogen, that the alpha carbon is chiral. And so that tells us that when it says here, except for glycine, all amino acids are chiral, that tells me that the structure of glycine must be this. I must have my carboxyl, my alpha carbon, and my amine. Because if glycine, if the alpha carbon is achiral, that means that I have two hydrogens here, and that would be absolutely correct. And so this is the structure of glycine right here. The simplest possible amino acid, glycine. There you go. All right. Well, a peptide, and I'm sure you've all heard the word peptide come up in maybe a biology class or introduction to biochemistry or something. Well, what a peptide is, it's a relatively short chain of amino acids, generally less than 40 or 50. And we use prefixes like di, tri, tetra, penta, etc. to describe the number of amino acids in a peptide. Say, for example, you have 3 amino acids, it would be a tripeptide. If you had 5, it would be a pentapeptide, so on and so forth. Whenever we throw out the word polypeptide, that just means that we have a chain of less than 40 or 50 amino acids. Once we get beyond that, then we're talking about proteins. Now, another term that's going to come up a lot in this chapter is a peptide bond. And the good news is that you already know what a peptide bond is if you were to see it, because an amide is a peptide bond, right? You are all familiar with an amide. If I asked you to identify an amide, you'd say, well, Mr. Guion, it's got a carbonyl, then you have a nitrogen, and it could have 2 hydrogens like this if it was a primary amide, but you all know what an amide is. Well, when you have an amide that links 2 amino acids, okay, those are called peptide bonds. So a peptide bond is nothing more than an amide bond that connects amino acids. Now, you can see that the amino acids are inside these green boxes, right? I have my carbonyl, my alpha carbon, and my amine. You go to the next one, I have my carbonyl, I have my alpha carbon, and my amine. But you see that they're linked together by these peptide bonds. And when we talk about protein, that means that the molecule is going to have some kind of biological function. And if you've ever wondered about what an enzyme is nothing more than a protein. So it's just a bunch of amino acids all linked together. However, it functions as a biological catalyst. So what are the amino acids? Well, it says here that there's 20 alpha amino acids that occur naturally, and they differ in their side chains or their R groups, right? If I draw the structure of an amino acid the way it was on the previous slide, we have our alpha carbon, then we have our amine, okay? And then we have our R group like this. Well, this is just taking that and drawing it as a Fischer projection, okay? So if I was to take this compound here, which is L-alanine, if I draw it as a Fischer projection, you can see that the amino group is on the left-hand side, okay? So when the amino group is on the left-hand side, that's an L amino acid. And we just saw in the carbohydrates chapter, the naturally occurring carbohydrates are D sugars like D-glucose, D-galactose, D-mannose, D-maltose, but the naturally occurring amino acids are the L amino acids. So their stereochemistry would resemble L sugars, but we didn't spend a lot of time talking about L-glyceraldehyde, for example. But again, if you look at L-glyceraldehyde, you see that the hydroxyl on the chiral carbon is pointing to the left. And so in L amino acids, the amine is going to be pointing to the left. Okay, so L amino acids and D sugars, these are the naturally occurring compounds that we are mostly concerned with as organic chemists. Well, you might be wondering, what are the 20 amino acids? Okay, what are the 20 alpha amino acids that occur naturally that we are going to be concerned with in this class? Well, let's take a look. I've kind of broken them up here as they're broken up in the table in your textbook. It says amino acids here that have nonpolar side chains. So something with a nonpolar side chain, you can see that they all just contain carbon and nitrogen except for one exception here, which is tryptophan. But let's take a look, okay? If you think of the structure of an amino acid, okay, they all have this in common, right? They all have the carboxyl, they all have an alpha carbon, and they all have an amine, okay? It's usually a primary amine except for proline, which is a secondary amine, but I'll talk about that in a second. Now, the R group is what gives the amino acid its identity. If you think about all the people in this class, right? If you were to compare our DNA sequences, we have a lot more in common than we do that makes us different, right? If you look at, I don't know, they say the DNA of a person in a cucumber is mostly alike. But anyhow, if you think about people, right, we all have a lot more in common than we do different, right? We have 2 arms, 2 legs, 2 eyes. We have a brain and a heart and, you know, blood vessels and a spine, whatever. But you think about what makes us all unique, and this sounds kind of cheesy, but it always helps students remember it. You think about what makes us unique, it's our personality, right? We all have a personality. And I like to think about the R group of an amino acid as kind of being the personality of the amino acid. It's what governs its identity. So again, you're going to see that this part here is going to remain relatively constant, okay? But the R groups are going to vary, and that's what gives the amino acids their identity. So take a look at all these different R groups. As I told you a few slides ago, that when the R group is just a hydrogen, that amino acid has no chiral center. That is glycine. Okay. If it's a methyl group, it's called alanine. And when you have an isopropyl group here, it's called valine. I always remember the isopropyl group. It kind of looks like the letter V, valine. Okay. Anyhow, but you also see that the amino acids, not only do we have their structures and their names, but we also have abbreviations. We have a 3-letter abbreviation. So this is the 3-letter. And then you have the 1-letter. Okay. Luckily, there's 26 letters in the alphabet, and there's only 20 amino acids. So everybody gets their own letter. Now, I don't expect you to memorize the 1-letter abbreviations, their structures, or the 3-letter abbreviations. If you were to need them for your quiz, I would give them to you. However, for my students who are biochemistry minded. If you're going to enroll in biochemistry in another semester, I've had many students contact me and say, hey, Mr. Dion, can you send me the list of the amino acids and their 3-letter and 1-letter abbreviations? Because in those classes, it'll be one of the first things you have to memorize. You have to know all of the naturally occurring amino acids, their 1-letter abbreviations, their 3-letter abbreviations, as well as their structures. All right, so let's move on from there. Valine gets repeated, I guess. Then we go to lysine. Lysine has an isobutyl group. So there's an isobutyl group on lysine. Then you go to isoleucine, which has a sec butyl group on it, which is kind of funny. And there's a chiral center on there, and you see that this methyl group is pointing down the way it's written. You can see the 3-letter abbreviations and the 1-letter abbreviations. I'm not going to go over all those. Next, we have an interesting one, which is proline. It is the only amino acid that the amine is tied into a ring like this. So it's not a primary amine, it's actually a secondary amine. And that actually gives proline some really unique characteristics. It makes it a very interesting amino acid. On the next one, you have phenylalanine. If you go over here to alanine, if you take the methyl and you just add a phenyl to it, then you've got phenylalanine. The next one is tryptophan. Okay, tryptophan, you can see it's got this ring here, this double ring system here that's aromatic. When you have an aromatic ring like this, so a 6-membered ring, and then you have this 5-membered ring on it like this has a name. It's called indole. Indole. And again, you don't have to memorize that, but there it is. So there's tryptophan, and I love the 3-letter abbreviation of tryptophan is W, which is not intuitive. It's kind of funny. All right, amino acids with polar side chains. So what are the polar side chains? A polar side chain means you've either got an amine as your side chain, right? Some kind of NH2 is part of an amide. So let me just throw that out there. Let me fix this. So you've either got an amide, come on, you, an amide, or you've got an alcohol or a thiol. Okay, those are considered the polar side chains. Okay, so let's take a look. So the first one is asparagine. You can see you get a primary amide here. You've only got one methylene there. The next one is glutamine, which has another primary amide, and you've got 2 CH2s here. If you're wondering about aspartic acid and glutamic acid, well, aspartic acid just has a hydroxyl here, and glutamic acid has a hydroxyl here. But we'll look at them on another slide. Next, you have serine, which has a methylene and then a hydroxyl. So that's kind of interesting. The next one is threonine, which has a chiral side chain. It's got this hydroxyl group on it here. Then you have tyrosine, which kind of looks like a phenol. It's like a benzyl with a hydroxyl over here. And the next one is cysteine. Okay, I always struggle with the pronunciation of this one because cysteine and cysteine are 2 different things. Cysteine is what results when 2 molecules of cysteine come together. So I'll call this cysteine, okay? And it's got that thiol on the end. The next ones are the ones that have acidic side chains, which is a carboxyl group. And then a basic side chain is something that contains nitrogen. So we have aspartic acid and glutamic acid, right? This is the acid analog of asparagine, which was up here. And then glutamine, its carboxylic acid analog is glutamic acid. Then for the basic ones, the easiest one I find to remember is lysine because it's just got a primary amine on the end. But then you have arginine, which has got this funky piece over here, and then histidine, which has an imidazole on it. So this is called an imidazole ring, this thing here. So there you go. Those are all the naturally occurring amino acids that we're interested in. Again, in Chemistry 3111, you are not asked to memorize them. However, it might be helpful for you, especially if you're thinking about taking biochemistry in the next few months. Something to think about there. So out of all the amino acids, all 20 of these amino acids, our bodies, like your body, my body, we can actually make 10 of 20 of them on our own. We can build them if we need them. However, the other 10, we can't make them. So that means they have to come from our diet. Now, the only time you're going to find a food that possesses all 10 of the amino acids that we cannot synthesize, and we call these essential amino acids, essential because we can't make them. If you want the 10 essential amino acids, you can get them from meat, okay, like meat, fish, milk and eggs as well. It says here there's no single plant source that contains all of the essential amino acids. For example, rice doesn't have all of them, corn doesn't have all of them, so you can mix them together to make succotash, in which case you would have them all. But I think that there is, I mean, you guys might want to double check me on this one, but I was wondering if soy might have them all. I'd have to double check on that. But anyhow, I mean, since somebody who's a vegetarian is not going to just eat soy all day long, so people who are vegetarians need to be careful to balance their diet to have a mixture of grains and beans and nuts and things like that in order to make sure that they have all of the essential amino acids. Again, if I've had many students who have come from the military and they'll say, oh, in the military, we would eat succotash from time to time. I don't know if they do anymore, but the reason why is because that's a good way to give people complete protein and it doesn't cost an arm and a leg, right? Rice and beans, cheaper than buying a T-bone steak. Did somebody have a comment about that? Okay. I thought maybe somebody was going to unmute their mic and be like, I love succotash or something like that. Anyhow, let's move on from there. Now, besides the alpha amino acids, yes, there are other amino acids, okay? We have like GABA, for instance, okay? That's a neurotransmitter, or thyroxine, which is a hormone that's found in the thyroid gland. You can see that these are amino acids. Any compound that has a carboxyl and an amine in it is technically an amino acid. It's not an alpha amino acid. These aren't anyway. Another example would be PABA, right? And that's para-amino benzoic acid. So that's this compound here where you have a carboxyl here, and then you have an amine over here. So this is PABA. But again, the same thing applies. You've got a carboxyl and you have an amine. So there are other amino acids out there. They don't have to be the alpha amino acids. And when you study biochemistry, this is just kind of an aside, but something that you'll discover is that there are more amino acids than the 20 that we looked at. There are others that do come up from time to time in biochemistry. Well, what about the acid-base properties of these amino acids? Because in the compound, we have a carboxylic acid and an amine. How is that going to react? Well, if you have your amino acid, I'm going to draw it out like this, okay? If we have an amino acid, okay, and if we put this at a low pH, let's say we put this at a pH that's equal to 2, that means it's very acidic, okay? Acidic exclamation point. Well, what's that going to do? It's not going to have any effect on the carboxyl, but it's going to certainly protonate the amine, right? The amine is going to pick up a proton, and then you're going to end up with an amino acid that's overall +1, right? It's got a +1 charge because you have a positive charge on the amine. Okay, well, that's fine. But each group in the amino acid, the proton that's highlighted in green, and the proton the blue proton highlighted in green, and the red proton highlighted in green, they both have their own unique pKa value, don't they? Right? In fact, the most acidic of the two of them is going to be the carboxyl hydrogen. Okay, the carboxyl hydrogen is the most acidic, and then at that point, once it's removed, you end up with a carboxylate and an ammonium. So you've got what's called a Zwitter ion, right? And a Zwitter ion is a compound where you have both a positive and negative ion in the same compound. Well, there's also some acidity associated with this, and the protonated amine could also be deprotonated to form an anionic species that has an overall -1 charge. So we go from +1, we treat it with a base, we end up with something that's neutral, and then we treat it with base again, and we end up with something that's -1. So as we go from a low to a high pH. All right, well, if you think about the pKa's of those two protons, it says here that the carboxylic acid, as I told you, is more acidic than the ammonium ion. Well, if you look at the pKa's of the carboxyl groups, okay, you see there, where's my highlighter? They're all in the same neighborhood. They all have a pK of around 2. Okay, so we'll say here pKa1 is around 2. And if you look at the pKa of the protonated amine, you can see that they're all kind of in the same neighborhood too. They're all around 9 to 10. Okay, so we'll say here 9, we'll say 9 to 10-ish. And again, take that with a grain of salt. Could be a little higher, could be a little lower. All right, so again, the carboxyl group proton is more acidic, and the amine proton is less acidic. Now you notice that the side chains, depending on your carboxylic acid, if you have arginine, okay, or maybe aspartic acid, which is a carboxylic acid, glutamic acid, you see that the side chains have pKa's associated with them as well. Well, we know that the pK of a carboxylic acid is around 5, and aspartic acid and glutamic acids both have carboxylic acids in their R groups, so it makes sense that their pKa's are around 4-ish, okay? All right. If we think about cysteine, its R group is a thiol, SH, has a pK of around 8. That makes sense, right? Thiols are more acidic than alcohol. So that's the kind of rationale that we want to take. All right. So there you go. So now we have the pKa's associated with the side chains as well. All right. So what have we covered so far? We've covered the carboxyl group as having a pK of around 2. We've covered the protonated amine as having a pK of around 9. And we've covered the side chains, which have variable pKa's. If it's an acid, it's going to have a pKa, that's lower than if it's basic. For example, tyrosine, it's got a phenol that's got a pK of around 10. Lysine, when it's protonated, it's got a pK around 10 as well. Anyhow, there we go. So what do we do with these pKa values? You might be like, okay, that's cool. Yeah, what to do? So you might be wondering, what do I do with these pKa values? Okay, say for example, we're looking at alanine. Okay, let me draw the structure of alanine for you. So alanine just has a methyl group. So I'll draw this. So we're going to draw it the same way that they had it on the slide back there. So I'll fix this. I'll draw my methyl group here. There we go. And this is the way I think they had it. Okay. So there you go. So there's alanine. Well, the carboxyl group, okay, the carboxyl group has a pKa of 2.34. So that proton is a pKa of 2.34. So it says here that when the pH is equal to 2.34, okay, the amount of protonated carboxyl and the amount of deprotonated carboxyl will be equal. And that's based off of the Henderson-Hasselbalch equation, right? The pH is equal to pKa plus the log of the concentration of the conjugate base divided by the acid. So if you take the log of 1, you get 0. So in that case, you get that pH is equal to pKa. So again, Henderson-Hasselbalch equation if you need to review it. But anyhow, so at a pH of 2.34, we have an equal amount of this form and this form. Well, what's physiological pH? Right? Physiological pH is around what? 7 point, I don't know, if I ask my students, I always get a different answer. It's usually, it should be around a pH of around 7.35 to 7.45, let's say. So physiological pH is more basic than the pK of that proton. So if the pH is higher than 2.34, that means that the carboxyl group at physiological pH is mostly going to exist in the deprotonated form, in the carboxylate ion form. So again, to repeat, at a pH of 2.34, you're mostly going to have the carboxylate, at a pH of 7.35 to 7.45, you're mostly going to have the carboxylate ion. Well, what about the amine? What about the amine? Well, it says here that at physiological pH, the structure of the amino acid is going to be Zwitter ionic. Why? Because what was the pKa of the nitrogen of most of the amines? It was around 9 to 10, right? The pKa of this was around 9 to 10. So if you're at physiological pH, right, it says here that physiological pH is higher than the carboxyl, but it's lower than the protonated amine. So physiological pH, again, let's just call it 7.4 for fun. So since this is more acidic than the pKa, that means that this is going to exist in its protonated form. So that means the take-home message is that at physiological pH, you're going to have yourselves a Zwitter ion, right? You're going to have the carboxylate ion, and you're going to have the protonated form of the amine, like this, okay? That's at physiological pH. Well, at physiological pH, amino acids are amphoteric. That means that they can function as either an acid or a base, depending on the situation. Here I have my Zwitter ion. It's all good. If you treat it with a strong enough base, like hydroxide, it's going to rip the proton off of the amine, okay? And the equilibrium is going to lie to this side over here, right? If you think about this as being the acid, let's say your pK is around 9 to 10. What's the pK of water? The pK of water is equal to 15.7. So the equilibrium is going to lie to the side of the weaker acid. Conversely, if you look over here, we have the carboxylate. If you treat it with a strong acid like hydronium, it's got a pKa that's equal to negative 1.7. Well, once it gets protonated, you form your conjugate acid. It's got a pK of around 2-ish. So again, the equilibrium is going to lie to the side of the weaker acid. So you have to know what happens to your Zwitter ion if you treat it with a strong base or a strong acid. You've got to be able to figure that out. Now, there's a property of amino acids called the isoelectric point. Okay, we abbreviate it with Pi, and that is the pH at which the Zwitter ion concentration is the highest. Okay, so the concentration at which the Zwitter ion, again, which is the form where we have the carboxylate and the ammonium at the highest concentration. So how would we determine what that pH is? Well, what we do is we look at the pKa values of the two protons, okay? Of the carboxyl proton and the protonated amine proton. We take the average of the two, and there you go. So any amino acid, and this is important, if the amino acid doesn't have an acidic or a basic side group, so you can leave things out like aspartic acid or histidine, for example, well, you just take the average of the two, and there's your isoelectric point. So at that pH, at pH is equal to 6.02, you're going to have the maximum concentration of the Zwitter ion. Now, what if you have an acidic or a basic side chain? What do you do in those cases? Well, I'm just reading off the slide here. It says for other amino acids, you average the pKa value of the side group with the matching amino side group. What does it mean by matching? Matching means this. If you have an acidic amino acid, you average the pKa value with the side chain of the side chain and the carboxyl. If you have a basic amino acid, you average the pKa value of the side chain plus the protonated amine. Therefore, you have to know what the acidic amino acids are. I'll give you a clue. They both end with the word acid, glutamic acid and aspartic acid. And for the basic amino acids, what are they? We go back here. We have lysine. We have histidine. So where are they? Back here. Here they are. We have arginine, histidine, and lysine. So you need to know the basic amino acids. You have the acidic ones here. So let's go back. Where was I? Somewhere around here. Okay, so here we go. So let's say you have lysine. Okay, well, that's got that basic side chain over here. So you take the average of those two pKa's. That's your isoelectric point. If you had glutamic acid, you take the average of the two carboxyl group pKa's, which are kind of hard to read. There you go. And there you go. There's your isoelectric point. And that's it. I bet you some of you who have studied biology have heard of the technique called electrophoresis. Electrophoresis is a way to separate amino acids by exploiting their isoelectric value. Now, how do we do that? Well, we evaluate what the isoelectric value is of our amino acid. And if we put it in an environment where the pH is lower than the isoelectric point, that means it's going to be in a more acidic solution. What's that going to do? It's going to protonate the amino acid, right? And you're going to end up with this form. You're going to end up with a cationic form, right? You'd end up with something like this, NH3+, okay? Well, if it's positive, if it's cationic, it's going to be attracted to a cathode where we have a negative charge. So say, for example, you have lysine. Well, lysine is a basic amino acid. so its isoelectric point is going to be pretty high. If it's at a pH of 6, well, 6 is more acidic. So if lysine is in a pH of 6, that means it's going to be doubly protonated, okay? It's going to be very positive, and therefore it's going to move towards the cathode. On the other hand, if you have an acid, right, something with an acidic side chain, okay, say for example glutamic acid, which has an isoelectric point of 3.22, well, if it's in a buffer that's at a pH of 6, that means it's in a more basic environment than the isoelectric point. So what would it look like? In that case, your acid would look like this. You'd look like this, where you have the carboxylate, okay, and then you'd have this. So that means it would be anionic. So if it's anionic, it's going to move towards the anode, which has a positive charge. Let's say you have aniline. Well, it's going to be pretty close to the pH, so it's going to exist as a zwitterion, but that would be a way to separate a basic amino acid from an acidic amino acid from a neutral amino acid, like that. Now, what would you do if you had two amino acids that have similar or very close isoelectric values? In that case, they will actually get separated by their masses, because the heavier amino acid is just going to move more slowly in the electrophoresis chamber. So larger means higher molecular weight. Amino acids just going to move slower. I mean, the whole concept of electrophoresis will probably be covered in more detail in biochemistry for anybody who takes that class. It might be worth the value of one multiple choice question for your quiz. All right. Well, you've probably, besides electrophoresis, which is an analytical scale technique, what that means is analytical just means you can use it to separate tiny, tiny amounts of amino acids. If you have a bunch of an amino acid, or if you have a big mixture, you wouldn't use electrophoresis to separate it. In that case, you would use column chromatography or a crystallization or something like that. Now, amino acids, since they're zwitterions, they share a lot of things in common with ionic compounds, right? Think about an ionic compound like sodium chloride. It's got a positive charge and a negative charge. It's positive on your sodium, negative on your chloride. Well, since an amino acid will exist as a zwitterion, they're generally speaking going to be colorless. They have high melting points. They have good solubility in water. But even though amino acids are generally speaking colorless, you can determine whether they are present or not by using ninhydrin as a color indicator. So as long as you have a primary amino acid, which excludes proline, so not proline, which has a secondary amine, but any other amino acid in the 20 amino acids, so 19 of them, will react with ninhydrin in the presence of sodium hydroxide to give you this purple colored product here, where the nitrogen from the amine is incorporated into the molecule. Then you're left over with some byproducts, which are shown here on the far right: water, carbon dioxide, and some aldehyde byproducts. Well, you've probably seen, or you might have seen, ninhydrin stain being used on crime shows and things like that. And the reason why they can use that is because people don't deliberately leave amino acids behind at a crime scene, but our sweat is composed of mostly water. The vast majority of it is water, but there are some amino acids that come out in our sweat, and so those can be stained, and that's how we can identify latent or invisible fingerprints at a crime scene. Well, with all that information in mind, it's always good to stop for a minute and try a practice problem. This is the first practice problem in the chapter. It seems like a good place to start. It says, although most naturally occurring proteins are made up of only L amino acids, proteins isolated from bacteria will sometimes contain D amino acids. Draw Fischer projections for D-alanine and D-valine. In each case, assign the stereochemical configuration as R or S at the chiral center. Well, let's start by writing out, I'm going to start by writing the natural amino acid where the amine is on the left-hand side. So if I want to write out alanine, so like this, I have my alanine, this is my R group, this is L-alanine, L-alanine. So if I want to write out D-alanine, what's it going to look like? It's going to be the mirror image. So I'm going to have my amino group over here, my carboxyl group here, and then my methyl group here. So this is D-alanine. And if we want to determine the stereochemical configuration, the amine has highest priority group. This is lowest. This is second highest. This is third. So I go like this. which is counterclockwise. My hydrogen is horizontal, and so I reverse that. And so the stereochemical configuration is R. So that means that this is R. If I look at the next one, which is D-valine, so D-valine is going to look like this. I'll put my carboxyl group at the top. Remember that the R group in D-valine was an isopropyl. I'm just going to draw it like this. If it's D, our amine is pointing to the right-hand side. So this is, again, an unnaturally occurring amino acid, D-valine. If I want to write L-valine, that's going to look like this. Oh, I guess that's what I wanted to draw, wasn't it? I wanted to draw D-valine. Anyhow, so D-valine is an unnaturally occurring amino acid. So if I want to determine the stereochemical configuration, I have 1, 4, 2, 3. So I go like this, which is counterclockwise. I reverse it. And so this is also R. There. So we know that naturally occurring amino acids are going to be S. There is only one exception. Okay? All naturally occurring amino acids are going to have the S stereochemical configuration. So we'll say so we'll say like this. 19 are S except cysteine. Okay, and there's a question that I assigned that deals with cysteine and enables you to, or helps you to practice and determine why cysteine is not S, but it is rather R in the natural form. Anyhow, let's move on from there. Let's try a question. It says here, draw the form of the amino acid that is expected to predominate at the stated pH. If I have alanine at a pH of 10. Well, going back to the table, so I'm going to scooch back here. So this is alanine. Where's alanine? It's right here. So I'm going to take the pKa of the carboxyl group and the protonated amine. So we go back here, make a mental note of those. And now let's scribble them in here. So the carboxyl had a pKa that was 2.34. And the protonated amine had a pKa that was 9.69. And so if I want to determine the isoelectric point, I would take the average of those, but that's not what I'm trying to do. I'm at a pH of 10. So I'm in a, let's see here. We'll call this more basic than both pKa's. That means that my carboxyl is going to exist as a carboxylate. It's going to be deprotonated. And it means that my amine won't be protonated, but rather deprotonated. And so this is the form of alanine that would predominate at a pH of 10. Now, what about proline? We look up the pKa of the carboxyl group of proline. It's 1.99. And the pKa of the protonated amine, which is only has 2 hydrogens attached to it because it's a secondary amine. In the unprotonated form, it's 10.60. Okay, so with those in mind, this is going to be the pH of 10 is more acidic, and it's more basic. Okay, so what does that mean? That means when I draw my proline molecule, since I'm in an environment that's more basic than the pKa of my carboxylic acid, it's going to be deprotonated. So I'm going to have the carboxylate. Let's draw the ring. There we go. Oops, I can clean that up a little bit. There we go. So normally it would be protonated like this, but I'm in a pKa that's more acidic, so it's going to remain protonated like that. And the last one is glutamic acid with a pH of 3. So let's give that a shot. My carboxyl group has a pKa of 2.19. So I'm in an environment that's more basic. So I'll draw the carboxyl group as being deprotonated. This is the nitrogen over here. And then I have my side chain over here. Let's figure out the rest. Then the protonated amine has a pKa of, what is it, 9.67. So I'm in an environment that's more acidic, so that's going to be protonated. So NH3 plus. And then my side chain, which is this one here, has a pKa that is 4.25. So I'm in an environment that's more more acidic, so it's going to be protonated, and there is the form of glutamic acid that would exist at a pH of 3. In order to solve the next problem, we're going to need a calculator, and we would need the table 25.2, so I recommend looking at that in your notes. It says calculate the isoelectric value for each, or the isoelectric point rather, for each of the following amino acids. So we'll start with aspartic acid. For aspartic acid, my carboxyl group, and I've already gone ahead and written down the data. The carboxyl group has a pKa of 1.88. The protonated amine has a pKa that is 9.60, and the side chain has a pKa that is 3.65. And so when we have an acidic amino acid, we're going to take the average of the two acidic side chains. So our isoelectric point is going to be equal to 1.88 plus 3.65. Then I divide all that by 2, and I end up with 5.53 divided by 2. Should only have 3 sig figs, so I get 2.77. So that's my isoelectric point. For lysine, my carboxyl has a pKa of 2.36. My protonated amine, what am I doing here? My protonated amine has a pKa of what? I can't read my own writing, 9.60. So the isoelectric point is going to be 2.36 plus 9.60 divided by 2, and I get 5.98. Well, the last one is lysine, and keep in mind that lysine is basic, and so you would take the average of the two higher pKa's, right? The side chain and the protonated amine. So I'll let you try that one in your own time. And that brings us to section 25.3.